Welcome to our fourth episode of the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the articles of the month, or other articles published in that month's issue of Diabetes Journal that we find particularly interesting. We also have a Sweet Talk segment of the podcast, which is more of an open forum discussion of additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darleen Sandvall, and I'm joined by my podcast co-hosts, Dr. Kirk Habiger and Dr. Kevin Williams. Good to see you both today.
Hey, Dar.
Hey, good to see you.
It's Friday. Happy Friday. A little more enthusiasm there, Kirk.
Yeah. It's rainy here and I should be more excited, but you're right. It's Friday.
Any big plans this weekend?
You know, we're making the annual pilgrimage to the beach for spring break, so
Nice.
wrapping stuff up here.
Kevin?
Yeah, look at the blue skies behind me.
Oh, that's not nice.
Kirk can be jealous.
Yeah.
That's not a blue sky, Kevin. That's
No, you
full
don't think so?
clouds according to Colorado standards.
All right.
All right, we should get to work. Kirk, why don't you tell us about the articles we'll be featuring this month?
Thanks, Dar. This month we have two great articles. The first is from the Kalamajski group at Lund University. And in this work, they characterize a human genetic variant that modulates PPAR gamma coactivator 1 beta expression and its subsequent effects on brown adipocyte function. In the next interview, we'll be talking with Professor Yuval Dor from Hebrew University. He's going to tell us about his group's work on cell type specific DNA methylation patterns in the pancreas and how methylation-based analysis allows for a more accurate measure of cell type composition in the human pancreas.
Awesome. And this month, we also have a great interview lined up for our Sweet Talk segment. Kevin, tell us what's going on.
Absolutely. And this week's Sweet Talk segment, Darlene's going to be joining us, and we have the honor of speaking to Dr. Carmella Evans-Molina, who was awarded the prestigious 2023 Outstanding Scientific Achievement Award by the American Diabetes Association. She's contributed an article for this month's edition of the journal. We'll be exploring Dr. Evans-Molina's pioneering work. I know I'm excited about today's episode, as this work has not only advanced our understanding of diabetes, but it's also opening up new avenues for risk stratification and treatment strategies.
Awesome. I'm looking forward to joining you for that. Let's jump in.
Our first feature article for the April 2024 issue of Diabetes is titled Human Genetic Variation at RS 1007139 Correlates with Adiposity-Related Traits, Modulates PPAR gamma coactivator 1B Expression, and Alters Brown Adipocyte Function. We are joined by the senior author of this paper, Dr. Sebastian Kalamajski. Dr. Kalamajski is an associate researcher at Lund University. Welcome, Dr. Kalamajski. Congratulations on your paper, and thank you for being part of this month's podcast.
Thank you for having me.
So we'd like to give you the opportunity to highlight anybody in your group or other authors that contributed to this work before we get started. So if you'd like to do that now.
Sure. So I should acknowledge the first author, Miwan, that actually did a lot of work. He worked really hard with this paper. And a number of other co-authors, Rashmi and Lina and Thomas, that collected data from an African cohort that we used in this study as well. And of course, our maybe more senior co-authors, Hendrik and Paul Franks, also contributed quite a lot.
Wonderful. All right. So now maybe can you briefly summarize your research program and how that program led you to this specific project?
Right. So I thought that we maybe start with the basics. So I think we all know that what makes people different, how much you are predisposed to disease, how much you respond to medical treatments. You can ascribe that to your environment, but also to your own DNA, right? To your own genome. And there are tons of observational studies, like GWAS studies, that would link specific genomic loci with specific traits, including type 2 diabetes and obesity, which is what we are interested in. But kind of the knowledge gap that those studies have created, in a way, is that there are not enough follow-up functional studies. I mean, studies that tell you what's so special about all those different genomic loci? I mean, what's the mechanism behind the disease, right? And this is where we come in. We are trying to find out why the different genomic loci associate with adiposity, for example, which is what we are looking at in this paper. And what we have seen so far is that it's often single nucleotides variation within those genomic loci that have an impact on maybe gene expression or on RNA splicing. And as a consequence of that, that can have an impact on your traits, right, on your body weight or on your response to treatment. And yeah, so that's roughly what we are interested in and how we work, basically.
Great, thanks.
So in this manuscript, you focus on this gene variant that modifies PPAR gamma coactivator 1B expression. And of course, PPAR gamma is a gene that many of us who study metabolism will recognize. So from a broad perspective, can you tell us about the results that led you to this particular gene?
Yeah, sure. So we are quite interested in genes that we think are involved in energy metabolism because they can be relevant for obesity and for metabolic disorders. And why we recently have studied like a number of common genetic variants in PGC1B as well as PGC1A, actually. And the particular genetic variant that we explore in our paper, it comes originally from an epidemiological study from 2014. It was a study on the diabetes prevention program cohort. And in that paper, they have looked at whether common genetic variants in PGC1B associate with the cardiometabolic traits. And what they have found was that one of those variants associated with subcutaneous adiposity after lifestyle intervention aimed at weight loss. And another interesting fact is that PGC1B is expressed quite a lot in mitochondria-rich tissues, including brown fat. And brown fat is kind of a hot topic at the moment as well. At that point, we were already working with the brown fat cell lines, and we were also working with CRISPR and allele editing using CRISPR. So we thought, well, we could perform CRISPR-mediated allele editing of that genetic variant in brown fat cells, and then we would see how the different alleles affect the metabolic phenotype of those cells.
That was a great overview of my next topic, which is really the methodology and kind of how you drilled down to some causal mechanism. So maybe you can go a little bit deeper and tell us about your work in these pre-brown adipocyte cells and the mechanisms that you've worked out.
Yeah, so the main hypothesis to test was to find out whether or not this genetic variant is actually an EQTL, and that was quite important. And in the paper, we actually demonstrate experimentally that the minor allele, or the G allele, causes an increased expression of PGC1B, and it does not affect the expression of genes that are in the vicinity of PGC1B, in the what we call topologically associated domain. And then we also looked at how this minor allele affects overall phenotype of the brown fat cells. Actually, we did not see that much of an effect on the ability of the cells to differentiate to brown adipocytes. But then when we looked at the phenotype that relates to energy metabolism, the minor allele conferred higher expression of mitochondrial proteins and the higher mitochondrial respirations. And then we did another interesting experiment. We exposed the cells to norepinephrine, which is normally released after cold exposure or exercise or stress. And we saw that the combination, if you will, of norepinephrine and the minor allele improved the cell's lipolysis and improved mitochondrial respiration, which is kind of what you want in a healthy, well-functioning brown adipocyte, right? So that's so altogether, it shows that the minor allele of PGC1B has an impact on energy metabolism, which is quite interesting, we think.
Excellent. So one of the interesting things about this paper is that, we've heard of the bench to bedside kind of research links, but this is more like a bedside to bench and kind of back. Can you talk a little bit about this allele? We're calling it a modified allele, I guess, because it isn't really increasing metabolic risk, but there are some population level differences in the occurrence of this variant. Can you talk a little bit about that? if you have any indication if that allele is leading to phenotype differences at the population level.
Right. It's a very good question. It actually points out a common issue we have with US data sets. And the issue is, you probably know that US data often comes from people of European ancestry, and it's like a predominant data sets, right? And data from people like from African ancestry is not that abundant, although it's improving, but it's not very abundant. Yeah. But anyway, so our genetic variant, so to speak, of interest has about only 1% minor allele frequency in the global population. So it's not very high, but it is more frequent in African populations. So the highest frequency of this minor allele that we found was about 7 to 8% in certain ethnicities in Nigeria. And we have so far managed to study this variant in a small cohort of women in Tanzania, where it seems to associate with adiposity, and this is what we show in supplemental data in our paper. But of course, we would like to study this variant in other cohorts, to replicate our findings as well, and I hope we will be able to do that eventually.
Do you think there's any opportunity for using this allele or maybe other alleles to direct people to specific types of metabolic treatment, obesity treatment, or type 2 diabetes treatment? Sort of a personalized medicine strategy, if you will.
Yeah, so one thing we should remember maybe that I don't think it's the only allele that will affect your metabolic phenotype, so to speak. But it, at least from the in vitro data that we have based on isogenic background experiments, it looks like you should be able to improve your metabolic rate if you expose yourself to exercise, for example, more, or if you go fasting, then you will maybe activate this the expression of PGC1B better if you have the minor allele. But that is something that we have to prove, with clinical trials eventually. It's not, yeah, we cannot really say that much more right now.
Yeah.
That's great. So I want to talk a little bit more about your work in the cell line and the minor allele, the GG allele. They seem to have a higher norepinephrine-induced basal mitochondrial respiration as well as lipolysis, correct?
Yes.
And then since these are our brown adipocytes, does that suggest they're better at thermogenesis? And if so, maybe you can tell us a little about the implications of that for the field.
Yeah, I mean, from the seahorse data so we use seahorse to measure proton leak after norepinephrine stimulation, and we actually tried to measure thermogenesis with like a thermo compound that measures the temperature in the cells, but it didn't really work out very well. But from the seahorse data, it looks like this potential thermogenic effect is quite prominent after norepinephrine in the minor allele carrying cells. Yeah, so what norepinephrine does is that it stimulates lipolysis, and that would sort of feed free fatty acids into the mitochondria where they are, combusted, basically, if we keep things simple. And yeah, and it looks like the minor allele cells are better at thermogenesis than the major allele AA cells.
Got it. Great. Thanks.
I'd like to give you an opportunity to share any other results that we didn't already talk about or go into more depth on any results that you would like to do that with.
Well, we don't have any other results from that study right now. I mean, there are kind of implications of those findings in the field that potentially that I mentioned previously, and it's about trying to set up clinical trials, genotype-based clinical trials to actually see if we can just see how well our in vitro findings translate to real life, to real human beings. So that will be like the next step, and that would be kind of exciting to do. And
Excellent. Any plans to introduce the allele into a mouse model and do some in-depth phenotyping that way?
not really. So yeah, so the thing about mouse genome is that they don't have that. So if you look at the locus where you find this human genetic variant, if you compare that locus with the mouse locus, it actually lacks about, I think, 70 base pairs of that locus, including the SNP that we are looking at, including the genetic variant we're looking at. So I don't think we can make a mouse model based on that. And it's a human genetic variant.
Yeah, that's tough to humanize.
Yeah.
Yeah. Yeah.
All right. Well, Sebastian, thank you very much for sharing your expertise with us today and your recent work on the molecular mechanisms associated with this human variant. I encourage the listeners to read the full article, as our featured articles are freely accessible online and are in the April issue of Diabetes.
Another feature article we'd like to highlight from the April 2024 issue of Diabetes is titled DNA Methylation-Based Assessment of Cell Composition in Human Pancreas and Islets. We're joined by the senior author of this paper, Yuval Dor, who is professor of biology at the Hebrew University in Jerusalem, Israel. Welcome, Dr. Dor, and thank you so much for being a part of the podcast.
Hi, thanks for having me.
So, Dr. Dor, we like to start these interviews out with a little bit of an icebreaker question. So I'm wondering if you can share with us a fun fact about science that you have rolling around in your head. Maybe it's related to your research, maybe it's not, just something you'd like to share.
Whoa! So for the audience, this was not planned in advance. So from the top of my head. All right. So the fun fact that makes me most excited for 30 years now is the fact that whales and the smallest mammal, the pygmy shrew, there's about 1 million-fold difference in size, and it's definitely genetically determined, right? Yet if you take their genomes, give it to a molecular biologist, he won't be able to tell who's the smallest, who's the largest animal. So there's something genetic there that determines something that is varying 1 million-fold, and we have no clue.
Very cool. I like that
That's really
one.
good. All right. First thing we want to start off is asking you to summarize your research program and maybe just a little bit about how that dovetails into the project that we're talking about today.
Right. So much of my lab's interest is in diabetes and understanding beta cell biology and why beta cells fail in diabetes and how they might be regenerating. And more recently, what is the basis of autoimmune diabetes in type 1 diabetes. But then another branch of my lab is studying epigenetics. That is mostly DNA methylation, how DNA methylation marks cells to determine their identity. And one place where these two fields merge is in the current project. So what we've done in this project is we reasoned that in many, many instances in the study of diabetes and pancreas biology, you want to know how many beta cells are there in that preparation of islets or in that tissue section, how many alpha cells, how many endothelial cells, how many delta cells, how many exocrine acinar cells or ductal cells. And this is really a fundamental question that any pathologist actually is asking every day in their histological analysis, but also for our study of biology. You want to know, right, if in type 1 diabetes there are more alpha cells or less, or has the proportion of any cell type changed? It's a very basic question, and this, of course, is being looked at by many people all the time. And the standard procedure for assessing, we call it cell composition in an islet preparation or in a pancreas histological section, is by immunostaining, right? You use, for example, if you want to know how many beta cells there are in a section, you stain with an insulin antibody and you count the number of beta cells. If you want to know how many beta cells were in that preparation of islets, which you use to measure insulin secretion, you measure the amount of insulin in that preparation. This is standard procedure, but there are many major problems with that approach. The problems, I guess the key problem results from the fact that the amount of insulin may change, may vary. For example, under circumstances where pancreas has secreted much of its insulin under high metabolic load, all kinds of pathologies or conditions, the numbers that you read may give you an artifact. And it's not a small thing. You may, for example, conclude that in a given tissue preparation, there are no beta cells. So that individual has lost his beta cells completely. This might not be true. There might be beta cells there. They're just not expressing your marker of choice, etc., etc. And this is really a basic problem in many fields of biology, and particularly in diabetes. So by merging these two aspects of biology that we study in the lab, we came up with the idea that there's a more fundamental mark of cell identity than a given protein that is expressed or maybe lost from the cell, and that is DNA methylation. And this is what we've done in this project. We say maybe a word about DNA methylation. So the genome, of course, is identical in all of our cells, right? But every cell type, not organs are conglomerates of cells. Every cell type is expressing only a small subset of the genes in the genome. And the reason this happens is because of epigenetics, and the most fundamental layer of epigenetics is DNA methylation. So DNA methylation marks for silencing all genes in all cells that do not need to be, are not allowed to be expressed, and is only the genes that should be expressed in a given cell type are, broadly speaking, demethylated there. So this really determines and marks cell identity in a very fundamental and stable way. So the idea is that beta cell identity is better determined by quantifying DNA methylation marks than by looking at the less stable and less reliable insulin protein marker. And so that's what we did. So in a nutshell, what we did in that project is that we determined the DNA methylation patterns of all cell types of interest in the pancreas, identified loci sequences in the genome that are unmethylated in beta cells and methylated elsewhere, or unmethylated in acinar cells and methylated elsewhere. These became our markers, and we developed a PCR sequencing procedure to extract DNA from a preparation and determine from analysis of methylation patterns in that DNA preparation how many beta cells, how many alpha cells, how many acinar cells were there. So it's mostly really a technical advance that can be used, I think, by many people in the field, but we also try to extract some biological novelties.
So that was a wonderful introduction to your strategy, and you covered the benefits over maybe pure immunodetection. I'm curious, there's a lot of studies now that are using single-cell RNA-seq. Can you maybe compare your strategy of methylation and how it may be better, or caveats to methylation as compared to looking at these cells using RNA-seq?
The key difference, I think, is not in the seq part of RNA-seq, but in the RNA part of RNA-seq. And there are very powerful techniques that allow us to determine all the genes that are expressed in a given preparation of RNA. And this has become so sensitive that one can go down to individual cells. And that's wonderful and very informative. But this tells you about what we call cell states. So what genes this population or this particular beta cell or this population of beta cells is expressing right now. And RNA is very unstable, and the genes that a given cell can express or not express are varying all the time. So by looking at gene expression, you can really say what these cells have expressed right now. And this might not represent cell types, because the same cell type can sometimes decide to express this gene or this gene. For example, if there's hypoxia or ischemia, a huge set of genes that are responsive to lack of oxygen is turning on. So RNA in general is telling you about the state of the cells, but not about their fundamental identity. While DNA methylation tells you something else. It doesn't tell you what genes are specifically active in the last several hours in the life of that individual cell, but they tell you what is the basic unmovable identity of that cell type. So that's something of different technologies that are used to ask different questions.
Absolutely. Hey, thank you very much for that.
So in this manuscript, you mentioned the concept of empty beta cells. As I understand it, these are cells that have lost insulin immunoreactivity, but can still be identified using other hallmark features of the beta cells. So can you tell us a little bit about how you found this and the implications of the findings?
So beta cells are eliminated by autoimmune attack in type 1 diabetes. Right? In recent years, we found several surprising things about this process. First of all, we thought that almost all beta cells were eliminated right at the time of diagnosis. Turns out this was mostly based on what happens in mice. When people started to look at pancreata from humans that were diseased shortly after being diagnosed with type 1 diabetes, turns out that only about half the cells are gone. And actually, there are many beta cells that have remained alive at the time of diagnosis. So there's stuff to work with. What if you could stop the immune destruction process at this stage, the individual can still go back to normal glycemia. And there are other features, interesting features in human beta cell biology that were discovered. And one angle that is still being studied is the possibility that there is a population of cells, of beta cells, that have survived immune destruction and are just invisible to us, and maybe also to the immune system, because they've stopped expressing insulin. Right? Not much is known about that. But first of all, at a really fundamental level, if your assay to count beta cells is based on immune staining of insulin, it is in theory possible that the beta cell would get rid of its insulin, you won't see it, and maybe also the T cells will not see it. So it's an interesting phenomenon. Maybe you can bring this back to life. So some people may call that de-differentiated beta cells, perhaps, or degranulated beta cells. There are suspicions that such an entity exists, and there's some evidence that indeed such cells exist in our body. For example, there have been reports on cells that express the gene of insulin. The RNA levels coding for insulin is still there, but the insulin protein is not there, right? So this gave rise to the notion of empty beta cells, but really not much is known about that. And we're lacking markers to assess the presence of such cells, right? Because if you only use insulin, then how can you? This is one potential application of this technology, because if you're looking at markers that are much more stable and immovable, once your identity as a beta cell is determined at the level of DNA methylation, this will not change forever. Big things have to happen for this to be erased. So it's a tool that can be used to study that. That being said, on the samples that we were able to assess using that method, we haven't seen evidence for a major process of this type. So how do you do that? You count how many cells you see in a sample that are expressing the insulin protein, and then what fraction of the DNA in that preparation contains the marks of insulin. And there's no huge discrepancy. So far, we don't see evidence for a major such process. This sounds like a small thing, but in the niche of beta cell biology studying diabetes, that's a big controversy that is leading to a heated debate. And there's no tool that can help us identify that. But the study of empty beta cells is still, an open and exciting field, because it is possible that under some circumstances such cells exist, and they represent a population of cells we actually don't know, either so badly injured that they're attracting more immune cells, inflammation, and actually triggering progression of the disease, or that they're so good at hiding that these are the cells that will remain alive for 30 years now. And because in people that die after living for decades with diabetes, in their pancreas we still see cells that express insulin and have escaped immune destruction for many, many years. And that's one of the biggest puzzles in the field. It's possible that these cells have spent many years hiding without expressing insulin. So what I'm actually saying is that it's still an exciting field of research. We don't have definitive results, but there's now a stronger tool to study this phenomenon.
What age do these methylation marker, like what age of the cell do these methylation markers turn on? Are you able to look at sort of the lineage, or as the cell develops towards maturity?
Yeah, so it's a good question. So DNA methylation, more than anything else, is a marker of cell differentiation. So when a progenitor cell becomes committed to become a beta cell in this case, right, at the time of that decision, one of the first steps taken is to demethylate the gene for insulin. And from that moment on, that gene can be expressed and actually will be expressed forever, and to keep it methylated elsewhere, right? This is really a fundamental decision to be taken at the very early stage of development when the cells differentiate. And this remains for your entire life. So unlike other markers, this is really stable. So it doesn't tell you what is the age. By the way, there are other markers. An interesting point. There's a whole growing field in epigenetics now that has identified parts in the genome that are methylated in an age-dependent manner, allowing you to tell, for example, from a small sample of a few cells, what is your chronological age with extreme accuracy, probably plus minus 1-2 years. And also there's the concept of epigenetic aging. I mean, we're taking a small sample of blood from you, and you're saying, oh, your chronological age is X, but your epigenetic age, your DNA methylation, looks like you're younger 5 years than your chronological age. And this has become something very interesting and exciting in epigenetics, and this applies potentially also to diabetes research. But we haven't done that. What we've looked at is at this very stable, boring, immovable markers of cell identity that allow you essentially to count with high accuracy and reliability how many cells of each cell type you have in a preparation. This, we hope, will find many applications in future studies of diabetes.
Very nice.
So I want to pivot a little bit here, right? We've been talking a lot about the beta cell, but there are quite a few alpha cells, especially in the human samples that you're studying, right?
We have more than mice.
Yeah. So any insight that your methodology uncovered on that population of cells?
Right. So first of all, we identified methylation markers of alpha cells and can use them to determine easily the same PCR reaction, how many of them we have. So it's a tool that is available. The main finding, which is consistent with previous observations using actually protein markers, is that actually in people with type 2 diabetes, we see they have a higher number of alpha cells. We saw that in our small cohort of pancreata from deceased patients that had type 2 diabetes, but this is consistent with some few previous publications. The biological significance of that is not clear to us, but it's a fascinating, I find it to be a fascinating fact or observation that still calls for an interpretation. I should say that in type 1 diabetes, autoimmune diabetes, there's more and more evidence for a role played by alpha cells, but it's not necessarily that their numbers change, as our assay might show. It's more that they're uninhibited, so they're overactive. So when an alpha cell is overactive, is secreting more glucagon, right? This instructs the liver to produce and secrete more glucose. So this actually adds fuel to the fire, right? The problem, there's no beta cells, so no insulin. So the liver is pumping out glucose. That overactivity of alpha cells will add to the trouble. So this regulation of alpha cells turns out recently to be another component in the problem with type 2 diabetes, type 1 diabetes, but as far as I know, this is because of their overfunction, not necessarily hyperplasia also.
Right. So I noted that insulin was one of the genes that was used as a marker for beta cells in your methylation, I guess, characteristic, right? But glucagon was not chosen as a marker of the alpha cell, right? Does that have something to do with the relative levels of methylation around the glucagon gene, or can you provide some more insight on that?
Yeah, it's a good point, and we're asked that all the time. Our approach to the problem is that you have, I should have said before that in a previous very extensive study, we've characterized the complete methylome, that is, the entire sequence of the human genome as present in each human cell type. It was a big project on all major human cell types in the body, isolated fresh from surgical material. So it was a big effort published in Nature a year ago. So this gave us a gigantic atlas or map where you see the entire genome in front of you with all the possible sites that can be methylated. And for each cell type, you can say, all right, these are the sites that are uniquely methylated or unmethylated in that cell type compared with everything else in the atlas, right? So we're interested here in the DNA methylation as a marker, right? So when you do that analysis, you don't really care about gene names. You care about sequences that contain or don't. It so happened that one of the best markers for beta cell identity is actually a specific location within the insulin gene that is unmethylated in beta cells and methylated elsewhere. The glucagon gene is, of course, expressed only in alpha cells, but in terms of methylation, it's not the greatest marker because there are cells in the intestine that also express the same gene and give rise to this GLP-1, glucagon-like peptide, right? What Ozempic is mimicking. And from other reasons, turns out that in the genome or the epigenome of the alpha cells, there are other locations that are the best, more specific markers that distinguish this from other cell types. These, by the way, are typically gene enhancers that control the expression of genes that are located far away. That's why it's not always easy to assign a gene name to a sequence that is demethylated specifically in a given cell type, right? In the case of insulin, it was really the insulin gene, but in the case of alpha cells, this is somewhere in the genome. It's unmethylated only in alpha cells. It might be a distant enhancer of a gene that is really making sense because it's expressed only in alpha cells, but we don't know that. But these are definitive markers of the DNA of a given cell type, including alpha cells. So
Great, thanks.
Nobody studies that GLP-1 peptide anyway.
Overstudied, yes.
Dr. Dork, I want to give you an opportunity to highlight any other results that we didn't talk about yet from your paper, and then maybe talk about the strengths and limitations of the approach.
Yeah, thanks. I would highlight one finding that we didn't discuss here, and this relates to an application that I mentioned briefly in the beginning of the discussion. So you have a preparation of islets from a cadaveric donor, right? And you want to assess how much insulin is secreted by these islets, right? Also, a fundamental aspect of in the study of hundreds of labs, right? So you easily you stimulate it with glucose, and you easily measure how much insulin is being released. But you have to normalize it to how many cells were there, right? So there are many ways to normalize, right? You can say, all right, I had 100 islets in that preparation. It's a bad way to normalize, because in this preparation there was small islets, and in these preparations they were contaminated with acinar junk, and in that preparation they were gigantic. Not good. You can then normalize to the total amount of DNA that you have in the preparation. Not good, because in that preparation almost all the DNA belonged to beta cells. In that preparation it was all acinar contamination, so not a good idea to normalize, right? What's most popular is to normalize the amount of insulin secreted to the amount of insulin that is present in the preparation. You lyse this, right? Okay idea, but not great, because maybe in that preparation the individual has secreted almost all its insulin, right? So very little insulin amount has remained in the cell. So if now you measure how much was secreted per the very little that has remained there, you get the illusion that that particular preparation is secreting gigantic amounts of insulin. You'll be misled, right? So none of these three methods is optimal. What you really want to know is how much insulin is secreted per beta cell.
Right.
Right. So you count the number of beta cells, and turns out that there is now a method. You just extract DNA, and you immediately can tell what is the number of beta cells based on, because every cell has just 2 copies of the genome, and that's it, right? So when we did that, we were able to normalize insulin secretion in what we think is a better way. And there was a surprising result. Turns out that if you measure, it is possible to isolate islets from people with type 1 diabetes, recently diagnosed patients. Most of their beta cells are gone, but with effort, you can still isolate small clusters of cells. And you call this an islet preparation for people with type 1 diabetes. And you can see insulin secreted from these clusters. And there are many ways to normalize, as I said. When we did it the right way, it turns out that most of the stuff in these clusters were contaminating acinar cells. Very few beta cells remaining there. When you normalize properly the insulin secretion to the actual number of beta cells there, turns out that insulin secretion from those islets from people with type 1 diabetes is within the normal range, is a decent secretion. So if you take the remaining islets that have escaped so far immune destruction in that individual, you put them in normal, decent conditions in medium, they function okay, which is an interesting phenomenon, right?
Yeah.
The defect is not inherent, right? It's caused by the hostile inflammatory environment. And that's a finding that came out from this study. And again, opens the gate for many, many studies. This was really, I should clarify, this was a proof of concept study showcasing a new technology. This can now hopefully be used by large studies that are being conducted. We're looking at many, many samples, and now if this becomes a routine, which is, then hopefully, more data, informative, will be accumulating. So that's one thing, that it's a tool to better normalize secretion. I would say that more broadly, the possibility, it goes beyond the pancreas and type 1 diabetes. More broadly than that, the possibility to scrape a piece of a slide, histological section, and at once in a digital PCR sequencing based approach that does not require any treatment with finicky antibodies and any expertise of a pathologist. You just do a procedure, PCR followed by sequencing using a technology that already exists in the vast majority of labs, and get a digital, highly quantitative result telling you in that preparation, this is the proportion of each cell type. I think this is broadly useful. For example, histological specimens from cancer patients, you really want to know how many cancer cells are there and how many immune infiltrating cells are there. To study that right now is cumbersome. You need to combine many, many antibodies. They don't always work, right? With this very stable generalized DNA methylation markers, you can at once give a quantitative assessment. So I think it's a broadly useful methodology, a quantitative approach to assessment of cell composition with many applications.
And I guess the one limitation is just the spatial, right? You don't have the spatial
You're
component, at least
absolutely
not yet.
right. Absolutely right. When you scrape a slice of tissue, you lose histology. Absolutely right. There are many applications where this is still useful. You're absolutely right that the technology, the most weighted for technology, I think, in our field is single cell spatial DNA methylation analysis. It's a lot to ask for, but when this becomes available, one would be able to identify the DNA marks of each cell type in their natural location. And I think that then we'll discover many new things. I'll just give one highlight, right? The entire world, our small beta cell world, is studying the concept of beta cell heterogeneity. This refers to what you mentioned before regarding single cell RNA-seq, right? You take many beta cells, you look at the transcriptome of each one of them, you say, oh, they cluster to 6 different clusters. There must be 6 beta cell populations. I'm not sure at all, because as I mentioned, it's possible these are the cells that felt bad on that morning. If you had sacrificed, fixed the cells 6 hours later, right? So I don't know. But if you are able to show that there are 6 clusters in terms of DNA methylation, there's no way they could switch. This must represent a stable, true beta cell type. So that's a big deal, because then you may start asking questions about subpopulations that are busy responding to glucose versus others that are busy responding to amino acids versus others that are highly resistant or sensitive to immune attack and cytokines. The whole world is opening. So this is coming soon.
Yeah.
So that will be an exciting event. And I think many people are looking at that, also our group. So I think this will make a big change in how we can interpret information on heterogeneity of populations.
Well, we look forward to interviewing you again when you have more data.
Yeah, so this diabetes paper was led by, I want to mention mainly two names, was led by graduate student Zeina Deraushi from my lab, and co-mentored by my colleague, Dr. Agnieszka Schindler, all of us at the Hebrew University of Jerusalem, with colleagues from Hadassah Medical Center, from University of Pennsylvania, and others. It was a group effort, and I'm grateful for them, and also for the funding agencies that support our work, mostly the Juvenile Diabetes Foundation and the NIH.
Thank you so much, Dr. Dor, for sharing your expertise and discussing your recent work highlighting the use of DNA methylation to quantify pancreas cell types. I encourage the listeners to read the full article in the April issue of Diabetes. Thank you, Dr. Dor.
Thank you very much. My pleasure.
Welcome to this edition of Sweet Talk. I'm Kevin Williams.
And I'm Darleen Sandoval. We are joined today by a very special guest, Dr. Carmella Evans-Molina. Dr. Evans-Molina is director of the Center for Diabetes and Metabolic Diseases at Indiana University School of Medicine. Dr. Evans-Molina was awarded the 2023 Outstanding Scientific Achievement Award by the American Diabetes Association, and a summary of her lecture is published in this month's edition of the journal. Dr. Evans-Molina, thank you so much for joining us today.
Thank you, Darleen. It's really nice to be here with you and Kevin to talk about the article and just be asked some questions.
Yeah, before we dive in, we'd like to start off with a little bit of an icebreaker question. Outside of science, do you have anything that you are passionate about? Is there some kind of a hidden talent or hobby you might have?
I don't have any hidden talents. I have several hobbies. I think I'm passionate about my family. So I really enjoy spending time with my family. I like traveling with them. I have 2 pugs named Bean and Tater Tot, or Tate for short. So I'm quite passionate about them. They're my favorite people. And as far as hobbies, it's not a talent. I wouldn't call it a talent, but I really enjoy music. And a number of years ago, after carting my children around to lots of musical lessons and piano lessons, I decided that I wanted to try to learn the piano. So I've been taking piano lessons for about 5 years, and I'm about as good as a 3rd grader playing the piano.
Better than me.
Yeah, exactly. All right. On more serious notes or more scientific notes, your work highlights the importance of calcium signaling as well as protein modifications, transport, and trafficking in beta cell biology. Could you explain to the listeners why you think calcium regulation is so crucial for understanding diabetes pathogenesis?
Yeah, that's a great question to start with. So, everybody who loves the beta cell knows that calcium plays a really, really central role in regulating insulin secretion and is a very pivotal part of glucose stimulated insulin secretion and the secretory response. One of my very first projects as a postdoc led me to trying to understand what the role of calcium within the organelles was, the acidic organelles of the secretory pathway. Became very interested in understanding how calcium within the ER not only regulates things like insulin production, but regulation of some organelle specific stress responses in the beta cell, like ER stress, and then how that might intersect with stress in other compartments of the beta cell as well. So that's really where we focused a lot of time and energy, is in developing tools to be able to look at calcium regulation within those compartments in the beta cell, and then trying to understand what pathways lead to dysregulation of calcium signaling there.
Yeah, I really enjoyed reading the article in this edition. You give some vignettes on your research program and how it developed over time. And I found your attention to the kind of serendipity that occurs in scientific research of particular interest. And some of the examples you gave, it was being in the right place at the right time, and others, it was kind of the fruit of the collaborations that were involved. However, it was also apparent that your own, recognition, acknowledgement to take advantage of those opportunities was pivotal in your career. I was wondering if you could speak to the listeners at various career stages, maybe a little bit about this.
Yeah. So I think one of the things that I learned very early in my career is that it's really important to learn new things all the time, and to challenge yourself. And I think in part that came from where I did my residency. So I completed my residency in internal medicine at Mass General Hospital. And when you graduate from medical school, there's all this anticipation because you've been a student, and when you're in medical school, they wear these short white coats, and all your attendings wear these very long coats. So as a medical student, you graduate and you're like, I can't wait to get the long white coat. And so I arrived at Mass General and everybody was wearing a short white coat. And I was puzzled by this, and I was a little bit bummed because I wanted this long white coat. And it was really clear. They said, we consider ourselves to be lifetime learners here. And that really stuck with me. And I think that as I've progressed in my career, I really enjoyed learning new things and bringing new tools into my research. And so I think that maybe has made me cognizant of when there's new opportunities and things that I could add to my research questions and repertoire. And one of my favorite sayings is that chance favors the prepared mind. So I think that while, yes, there's been a lot of serendipity, what I consider to be serendipitous opportunities in my career, also I've been kind of receptive to observing new things or to bringing new things into the fold, and just really gotten a lot of enjoyment and passion from that.
You pioneered research into the role of the sarcoendoplasmic reticulum calcium ATPase pump, or SERCA pump, in beta cell function. So, can you tell us a little bit more about this pump system? Maybe summarize some of the details that your lab has discovered along the way?
Yeah, absolutely. So actually, I didn't even know what that was, what the circuit pump was, and it was a reviewer of this second paper that I wrote while I was completing my postdoctoral research at the University of Virginia when I was studying with Raghu Miramira there. So we had this very interesting calcium oscillatory phenotype in these DB mice that had been treated or not with pioglitazone. And the reviewer actually suggested that I look at expression of the SERCA isoforms in the beta cell. So we did that and made some interesting observations. And so when I was starting my lab, I thought, well, I want to understand why SERCA expression is dysregulated. And then I want to understand what happens when you knock it out in the beta cell, or you have conditions where either the expression or the activity is impaired. And so we've generated a number of different mouse models to be able to study this. And essentially, sort of key findings from that research show that if you impair calcium in the ER. So if you have reduced calcium in the ER, you have reduced insulin secretion, but there's also activation of ER stress signaling in the beta cell. And then more recently, we've been really intrigued with the observation that if you have reduced SERCA expression, which reduces ER calcium, but something I didn't quite appreciate until later in my career is that it actually regulates calcium in the cis-Golgi as well, in some of those intermediate compartments between the two organelles. And there's a disruption in protein trafficking. So that's been where we've been focusing our attention recently. And I think it's really interesting in the beta cell. A lot of people think about the ER, and a lot of people think about what happens at the plasma membrane. We know much less about what's happening between the ER and the Golgi and from the Golgi to the plasma membrane. So that's been something we've been really interested in recently in some of our projects.
Very interesting.
I will add that one of the really rewarding kind of translational research questions that we've been recently interested in is that there are people who have SERCA2 haploinsufficiency. SERCA2b is the most highly expressed isoform in the beta cell, and they have this condition called Dareen-White disease. It's an ichthyosis disorder, so it's primarily a dermatological disorder, but there's been a lot of recent interest in trying to understand what conditions these individuals have other than the skin phenotype. And so that's been a really interesting collaboration with the dermatologist to try to understand some of those phenotypes.
So an example, we always complain about our reviewers, but sometimes they can have a positive impact on our work.
Yeah, absolutely. I tell all my trainees, I don't know exactly who to attribute this quote to, but it goes something like you build your house out of the bricks that are thrown at you. And so I think that we always assume that reviewers are just kind of being mean or picking at us or holding us up, but there's always good nuggets in a review. And I think if we can kind of put aside our egos sometimes and just think about that as constructive feedback, that we can often really improve our papers. I've never sent back a revision that I haven't thought, well, this is a better paper because of this process I've gone through. It's sometimes painful, but it's good. It's a growth process.
Thank you. Kevin?
You touched on this a little bit in the previous answer to Darleen, but I was curious, as a clinician scientist, you're really sitting at a point in your research program that it has translational applications, clinical applications. And for our listeners, I was wondering if you could expand on any potential ongoing or maybe planned clinical trials or how you might envision the work ongoing in your research program being translated into the clinic.
Yeah. So I think when I think about kind of the progression or tangents that my career has taken, one thing that I'm really very happy that I did at the beginning, because I really get a lot of joy and passion out of cell biology, is I'm glad that I focused a lot of effort there in the beginning. And I didn't even know how to do basic science until I was a clinical fellow at the University of Virginia, and I started working in the lab. But I spent a lot of time really trying to hone those skills and learn how to do basic science. And I just, love cell biology. But then along the way, I was given opportunities to get involved in some translational research projects, clinical research projects. And that was has been and still continues to be a really steep learning curve for me. But I was given the opportunity also to participate in something called the Type 1 Diabetes Trial Network. I was invited to be a member of the team at IU by Linda DeMaglio, who's been just an incredible friend and career mentor to me. I started going to these meetings, and I, started out kind of very gingerly being involved in some of the projects and the clinical trials. But over the years, I saw this intersection of how my basic science might intersect with some of the things that I was doing at TrialNet, and that's been really rewarding. That's also led us to be interested in how we might use some of our knowledge of beta cell stress to develop biomarkers. But then very recently, I was given the opportunity to be a study chair for a trial called the Jackpot trial. And in this study, we're going to be testing 2 JAK inhibitors in stage 3 type 1 diabetes. That's been really fun because it dovetails with some of the stuff that we do in the lab. We've been testing compounds that interfere with signaling through some of these cytokines that signal through JAK and TYK2. And so this is to me, it's been a great learning opportunity to write the clinical protocol, get the clinical trial up and going, but then also, again, to marry concepts in my basic science with the clinical research as well.
Yeah, that's really cool. To have more sort of bigger picture impact on the field, if you could give one piece of advice to young scientists entering the field, what would it be?
Gosh, it's hard to think of just one.
You can have multiple if you'd like.
So I have an opportunity to work with a lot of clinical trainees, and I am always really trying to convey to them what an amazing opportunity it is to be a physician scientist. And I really worry that we're in endocrinology and diabetes research, that we are losing an opportunity to retain people who could be thinking about it from the patients that they see in clinic and the important research questions that are informed by their clinical practice. And I don't know how to stop that loss of this brain power from the research community. So I think that for me, it's always, as a physician, it is a path of comfort. You know clinical medicine, and you know it like the back of your hand when you finish your training. And so it feels very comfortable to have this great mastery of how to be a physician and take care of people in the clinic. It is much more anxiety provoking to think about, how can I keep myself funded? How can I think of research questions? How can I mentor people and be successful in this pathway that seems very nebulous? So I think that if I could put that all into one sentence, I would say, don't be afraid to take a chance and bet on yourself, and really think about the things that give you passion and design a career around that. And so that would be my advice.
I like it. I am a big proponent of focusing on the joy, right?
Yes. Yeah, absolutely.
And maybe one last kind of fun question. There is a lot of pomp and circumstance surrounding winning one of these awards at the ADA. What was that experience like for you? Can you share any funny or interesting stories about that process?
It is an incredible whirlwind. There's a lot that goes on before the meeting. And so there's this kind of pressure to make this presentation that, you've got to highlight in 30-ish minutes your career, and, worry about, did I acknowledge everybody who has had such an incredible impact in my career. So it's just there's a lot that goes into it. And then the thing that I think gives everybody the most anxiety, and it certainly gave me anxiety too, is this teleprompter issue. So they want you to write out every word that you're going to say. And then they're like, you can't change it after a couple weeks. And then, now you have to stand in front of this podium and just give a talk with a teleprompter, which is something we don't do as scientists, right? We like to talk and teach when we're giving lectures. So that was a bit of anxiety, but I would just say that it was such an incredible experience because, well, first of all, you go to the meeting and everybody's happy for you, and you get to bring your family. And so my husband and my 2 daughters came with me, and just kind of seeing their face in the audience as they listened was so cool. And they barely pay attention to you. I have 2 teenage daughters. And so one of my daughters was like taking notes through it. And it was just really, really just an incredible experience. So I got to share it with my family. I got to share that experience with close friends. And then we had the amazing after party with karaoke. So that was a highlight as well.
And the photo booth.
Oh, and the photo booth. Yes. Yeah. So I really couldn't have asked for a better meeting. And
well, maybe if Bean and Tate could have been there.
Yes, that's the only thing that was missing were those 2 pugs who are like the best anti-anxiety medications ever. These dogs that just like to snuggle with you.
Well, thank you, Dr. Evans-Molina, for joining us today. To our listeners, thank you for tuning in to this edition of Sweet Talk. Be sure to check out Dr. Evans-Molina's article that she contributed to the April issue of the journal titled The Ailing Beta Cell in Diabetes, Insights from a Trip to the ER. And don't forget to like and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss each month are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you next month.